This is an electron micrograph at the level of the glomerulus. As you know, the glomerulus is a knot of capillaries, and in this knot, this is the endothelial side and these are the endothelial cells, and in between the endothelial cells are the fenestrations through which the fluid and molecules pass. And now we have got the basement membrane here, and there are several diseases in which the basement membrane is affected and the basement membrane is thickened, so that the glomerular filtration and the renal function are reduced. Now, on the capsular side or the Bowman's capsule, Bowman's space side, we have the food processes here, and these food processes are lined by cilia, and these cilia contain glycosaminoglycan coat and the glycosilial protein. And the filtration occurs through the slit diaphragms which are present between the food processes on the capsular side of the Bowman's capsule. So, the fenestrations here, the slit diaphragms there, and the basement membrane, and the fluid and molecules have to traverse. Whatever there is in the capillary lumen, all the smaller molecules and water are freely filtered, but plasma proteins are withheld here, they are not filtered. That is why all those important hormones like thyroxine, cortisol, or any other hormone for that matter is protein bound, so that the hormone is not filtered and then wasted in the urine. In order to save itself from filtration, there is strong protein binding of the hormones and other important substances. This diagram summarizes the transporter sites in the nephron. So, after the glomerulus, we have the proximal convoluted tubule, the descending limb of the loop of Henle, the loop proper, the thin portion of the ascending limb, the thick portion of the ascending limb, the distal convoluted tubule, the collecting duct. This is a schematic representation. Sodium chloride and water, when they enter into through the PCT, through the action of the ACP1, that is aquaporin 1 channels, water is passively diffusing into the interstitium here. Here also there are ACP1 channels, free flow of water occurs. Then there are the urea transport channels here, urea transport A2 channels, transporters. Then again ACP1 channels in the descending limb of the loop of Henle for free transport of water into the interstitium. The sodium chloride goes up here, and then again further loss of sodium and chloride in this part of the thin portion of the ascending limb of the loop, and then again here there is an active loss of sodium and chloride along with potassium into the interstitium, and this is the thick portion of the ascending limb of the loop, and then we have got the distal convoluted tubule, wherein again sodium and chloride and passive diffusion of water using aquaporin channels 2 and 3. Now, urea also diffuses here, and then we enter into the distal convoluted tubule, continuing into the collecting duct, and urea is transported out by the urea channels, and aquaporin 2 and 3 under the influence of the ADH will reabsorb water here. Here also aquaporin 2 to 4 and again aquaporin 2 to 4 in the inner medulla, and this part of the water transport is under the control of the ADH, and this is the outer medulla and this is the cortex. This schematic representation is taken from Journal of American Society of Nephrology, JASN. The proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule and the collecting duct. This part is highly permeable, the proximal convoluted tubule, to water. The descending limb of the loop of the Henle is also highly permeable to water. Then the thin portion of the ascending limb and the thick portion of the ascending limb are not permeable to water. Only sodium and chloride and potassium chloride diffuse through this part. Distal convoluted tubule, it permeates both sodium chloride as well as water. The collecting ducts, here the action of aldosterone comes into play, sodium reabsorption takes place in exchange for potassium, and then there is the water reabsorption through the aquaporin channels under the influence of the antidiuretic hormone. Remember the countercurrent multiplier system, which increases the tissue osmolarity in the surrounding tissue, the interstitium around the nephron, particularly in the medulla around the loop of Henle. And this is the one which allows the movement of water very freely into this because of the high concentration gradient, and also the moment of sodium chloride in this region is aided by the lower osmotic gradient here available compared to the more inner part of the medulla. Look at this diagram very carefully. This is the collecting duct. The final concentration of urine, final removal of water to reduce the urine volume to around 1 to 1.5 liters a day. 180 liters of fluid is filtered here at the glomerulus per day. The osmolarity is 290. Sodium and water are lost here. It is reduced to 36 liters per day by the time it enters the descending limb of the loop of Henle. Here further water loss, further reduction in the volume, and from here sodium is removed and the fluid at this level is 24 liters per day. The osmolarity of the fluid is hypoosmotic compared to the plasma, 60 milliosmoles. Further loss of solute, and then in the collecting duct further loss of water, and finally around 15 liters of fluid is presented to the collecting duct, and these 15 liters from 180 liters the fluid here is brought down to 15 liters, and urine has to be reduced from 15 liters to 1 liter per day or 1.5 liters per day, and this is what is the role of the collecting ducts. And here is important action of the antidiuretic hormone under the influence of the ADH, they are also called the vasopressin. The aquaporin channels transmit the water, and this is an enlarged view of the principal cell of the collecting duct. The principal cells of the collecting duct are very tight, they have very tight junctions for water. There is a vesicle of the aquaporin channel inside. You see the four-membered vesicle. What happens is, when the arginine vasopressin comes and touches the other end, the outer end of the collecting duct, like here, it initiates and gets attached to the V2 receptor on the outer end of the collecting duct principal cells. That triggers a mechanism for the production of cyclic AMP, and the cyclic AMP triggers the exteriorization of the aquaporin 2 channels. The aquaporin 2 channels which are available within the cell as a vesicle are translocated into the basolateral membrane side or to the tubular lumen side of the cell, and the aquaporin channels form conduits there. Freely water moves to this channel. The water that enters the cell now through the channels called aquaporin 3 and 4 moves out of the principal cell into the interstitium, and that is how the water is reabsorbed. So basically, the arginine vasopressin getting attached to the V2 receptor triggering the cyclic AMP, cyclic AMP triggering the vesicle form of the aquaporin 2 channel, translocating the aquaporin 2 channel onto the lumen side of the tubule, and wherein which the water freely enters the cell now, and this entered water is excreted into the interstitium through the action of the aquaporin 3 and 4 channels. So in nutshell, the arginine vasopressin, or also known as the antidiuretic hormone, in the company of the aquaporin 2 channels gains the water into the cell, and through the aquaporin 3 and 4 channels excretes it into the interstitium, and finally reduces this 15 liter volume of urine to 1 to 1.5 liters by the time it reaches the renal pyramid, through which it passes into the ureter and then onto the bladder, urethra and outside. We have already seen the animation of the aquaporin channels. Here is the cell membrane, and within the cell membrane we have the protein coils which form the aquaporin channels, and this is the water channel through which water movement is facilitated. The movement of water across this channel is several thousand times faster than the movement through any other part of the cell membrane. That is how these are specialized conduits for the transport of water. And in the distal convoluted tubule and in the collecting duct, precisely in the collecting duct, the aquaporin channels are under the influence of ADH or vasopressin and then conserve water and concentrate the urine. We already know that there are several aquaporin channels, as aquaporin 1, 2, 3, 4, 6, 7, 8, and to date there are 10 aquaporin channels which are already documented. They have several functions other than the function in the nephron. In other cell membranes also, aquaporins do act for the movement of water into and outside the cell. Look here, as far as the renal handling of the solutes are concerned, there are three types of curves that you can see. The one that you see on the top is the type A curve, and then the type B curve and type C curve. And here this is 100% glomerular filtration and renal function. And as the renal function starts decreasing from 100% to 70% to 50% and 20%, there is no uniform increase in all the types of substances. Type A solutes increase quite very fast even if the GFR comes down to around 50%, and from then onwards they are in the abnormal range. The example for that is type A solute creatinine. Whereas substances which belong to the type B class, they do not show any increment in the blood until and unless the renal function falls to around 20%. Unless the GFR comes to 20% or less, we will not see an elevation in these substances. Example, potassium, hydrogen and urate. Whereas solutes which belong to the class of type C curve, they never are shown an abnormal quantity in the plasma despite the renal function being as little as 4 or 5% of the normal. Sodium and chloride represent this type of solutes. So, if we are assessing renal function, it is important for us to use those substances which show up their abnormal values quite early in renal disease, such as creatinine. And there is another substance called cystatin C, which is much more sensitive indicator of decreasing renal function. Even when the renal function is around 20% or 25%, the normal, cystatin C starts appearing here even before the type A curve. So this is a very important concept for us to understand when we measure different substances in the body fluids, particularly in the plasma, and try to extrapolate that to the degree of renal dysfunction. Let us now see the role of ANP on the metabolism of sodium and water in the body. ANP stands for atrial natriuretic peptide. When there is an increase in the blood volume, it causes increased stretch of the atrial muscle. This increased atrial muscular stretch, the myocardial cells in the atrium release the atrial natriuretic peptide. This atrial natriuretic peptide has got action on the hypothalamus, on the kidney, on the adrenal cortex and on the medulla oblongata. By the action of ANP on the hypothalamus, there will be an inhibition of the ADH or vasopressin. This results in increased sodium and water excretion, and that reduces the blood volume. Now, when the ANP acts on the kidney, there is a twofold effect. One is the increase in the GFR, the other is the decrease in the renin. Both increase the sodium and water excretion and thus reducing circulating blood volume. The action on the adrenal cortex, it inhibits the aldosterone, thereby it facilitates the excretion of sodium, and along with the sodium water also goes out and there will be reduction in the blood volume. So, the action on the medulla oblongata directly decreases the blood pressure by vasodilation. That is how the ANP has got multifactorial action on different levels to control the increased blood volume and thereby adjust for sodium and water balance in the body.